guys. My name is Emery and I did environmental science at CU Boulder in the Schmidt lab. And so basically what I was researching was soil microbes. I did a lot of background research, more specifically on arbuscular mycorrhizal fungi, which basically it sort of sticks onto the roots of plants and helps the plants get vitamins and minerals from the soil, while the plant helps it by providing energy from photosynthesis. And I also did a lot of research on Roundup and the different uses, which obviously include agricultural to get rid of weeds and invasive species, and also habitat restoration. And some of the environmental effects of Roundup is basically nitrification of the soil, which, if that leads to runoff, can cause issues with waterways. And so I was doing follow-up research on an experiment that was done 10 years ago, in which they added different treatments of Roundup to a plot of land. Okay, so the first part of the experiment was the field work, in which we went back to that plot of land, and basically on each field we would take 2 to 3 plants from the different treatments and put them in bags so we could take them back to the lab. And then, so for the dyeing process, we were dyeing the roots so we could see all the little microbes when we looked in the microscope. And we had to start by bleaching all the roots to get any dirt and stuff off so we could see clearly when we put them in blue dye. And that process took a really long time, especially cleaning the roots. That took 5 hours. All right, microscopy. So this was towards the end of my internship, and I spent 3 days doing this and collecting data, and it was really straining on the eyes. And basically, we would look into the slide and move it around 100 times to get 100 different views and count the amount of fungi we saw on each slide. And if you look in that little circle right there, those little sort of darker dots in there, those are the fungi. We didn't really get to see the results, but I got to calculate a few data points. And up here you can see the AMF is the arbuscular mycorrhizal fungi, and with 0 applications of the herbicide, there's a higher colonization of the root. And once you get farther along with more applications, the root colonization decreases. So it's kind of harmful to the soil microbes. All right, and then this is important for the future of both agriculture and habitat restoration, so we can keep our soils and our plants healthy while also getting rid of invasive species and weeds. All right, so what I learned is the reality of working in a lab. It's a lot of desk work and just sort of analyzing data. It's not all field work and doing crazy experiments, but it was still very fun. And also how to present data in the correct way. I did a whole presentation at the end of my internship to everybody in the lab. And then also what it's like to live on your own. So I was living with Adriana, who presented earlier, and we were in student housing and we didn't have a host family, so that was an interesting experience. And then my advice for future interns would be to take initiative and go after what you want to learn about, and to ask a lot of questions because you're going to be confused a lot. And then to also be open to all opportunities given to you, even if it's not what you had in mind when you came into it. All right, so during my free time, I hung out with Adriana and I explored the campus. And then Stefan and Anna from Avery's presentation actually came and visited me, which was really fun. Spent some time with them. And yeah. And I'd like to give a special thanks to Sarah and everybody in the lab, Pacifica, who was my mentor. And yeah, thank you guys.